So I am Roger Legrand. My background is veterinary medicine. Then I did a PhD in virology and immunology, and then I started working here on AIDS vaccines and models for assessing the efficacy of these different AIDS vaccines. And this had been started about 20 years ago with a group led by, at that time, Professor Dominique Dormont. Our contribution to CHAARM is in two different aspects. The first one is to better understand mechanism of transmission of the viruses using the non-human primate models for that. And the second aspect is to assess the efficacy of different candidates developed by the partners within CHAARM. So far, I think our major breakthrough is to really identify and characterize cells in the semen of the animals that are infected with the HIV and can transmit infection and possibly transmit infection during the sexual contacts. I think that this is one of the few studies that demonstrate that semen can be infectious not only through the free viral particles present in the semen, but also through the infected cells that are present in the semen. And if we confirm that these both kind of viruses or infectious materials we have in the semen are relevant or can transmit really infection, it is very important that we develop vaccines or microbicides that can affect these two kind of infectious materials. Scientific part for understanding the mechanism of transmission, and we will also starting new big pharmacokinetic studies of combination of antiviral drugs used as microbicides. We don't have the results yet, and that will become soon. So the next year we will be able to discuss the results obtained in the animals, and that will help the design of the future trials, either in the animals or the clinical phases. The major issues we have to face for preparing these PK studies is more on the formulation part and how then once we will put the compounds into the vagina of the animals, how to collect the samples in order to preserve the drugs and to make the best extraction of the drugs we can to really measure what is in the fluids and the tissues exposed to the drug. So there were several exchanges with the partners for having the appropriate standard procedures. So we have achieved now 2 SOPs on which all we agree that will be used in the PK studies that are performed at the moment.